I will use the first case I'm going to show you to bring some more confusion into the question of corpus callosum and commissural agenesis. It is really very bright here. Can we turn down the lights? Thank you very much. A little bit more, because otherwise you won't see the images. Well, commissures on MR images. What do we see? We see the anterior commissure, the rostrum of the corpus callosum, the genu, the body, the splenium. We also see the fornix, and we see the posterior commissure. The commissure we don't see in normal cases is the hippocampal commissure, because we only see it under certain circumstances. So what is a commissure? Commissures are white matter bundles that connect analog structures on both brain sides. Commissures are thought to lead from one hemisphere to the other on a genetically determined path with a certain thickness. And so we don't have only the corpus callosum. And to add some more confusion, the nomenclature of radiologists is a little bit different, probably from that of obstetricians. Corpus callosum agenesis, it is missing completely. Hypogenesis, it is missing partially, and dysgenesis, it has an abnormal shape. First case, gestation week 22, there was an asymmetric ventriculomegaly, and the question was, what about the corpus callosum? Here you see some MR images, T2 weighted, then an echoplanar sequence to look whether there was hemorrhage in it, T1 weighted images, but here the T2 weighted images are sufficient to show, yes, something is there that could be a part of the corpus callosum. Then there is the unilateral ventriculomegaly, and then there are some septa. We don't know why they are there. So what did we do? Of course, we performed tractography. And what did we see? We saw bundles on the right and the left side. In green, you see the corticospinal tract. And I tell you, the colors of the bundles is chosen deliberately. So we cannot know whether these are two distinct bundles or whether there is a bundle going from the frontal lobe of one hemisphere to the occipital lobe of the other. And here you see it in a movie, how it looks like. And maybe there are two bundles on the right and the left side, but also it might be the case that there is a bundle shaped like an S. And this bundle has been described as the so-called sigmoid bundle. Well, it has been, but let me at first show you the case. Later on, it got unilateral ventriculomegaly because of a blockade of the foramen Monro, and we proved that using MRI. This is not really interesting. We were not able to do another tractography prenatally, but we could do it postnatally, and here we could prove the presence of this so-called sigmoid bundle. So what is this? It has been shown in animal studies, and it has been shown postnatally. And this is, but it had never been shown in utero in vivo. It was defined by Tovar Moll the first time in 2007 as a bundle that connects the frontal lobe with the contralateral occipital lobe. And what he wrote is whether the function of such bundles is compensatory or a maladaptation is a matter of speculation. In the meantime, we have collected cases, and we know it is rather, the function is rather maladaptive, and these bundles are not really functional. That means the prognosis of children, of fetuses having this might be not good. But let me come back to that later. To explain why we are interested in these bundles, I have to go back. I have to go back more than 100 years, I'm afraid, to a guy named Probst, because he was the first to describe a strange bundle in a brain that had no corpus callosum. And he called this the Balkenlängsbündel. It was defined as a striking and particularly interesting structure of brains with complete absence of the corpus callosum. And fortunately, this bundle became very prominent and famous because otherwise you would still have to remember the conception of Balkenlängsbündel, which is not so easy if you're not German speaking, but in the meantime it is of course called the Probst bundle. So why were we interested in this old work? Because the next thing he said was the absence of the corpus callosum is rather a heterotopia than an agenesis. And you know, of course, from gray matter, a heterotopia is a normal structure in an abnormal location. So the Probst bundle is a normal structure in an abnormal location. So it is the corpus callosum in an abnormal location. First step to prove it was to correlate our findings with histology. And there you are. Here you have the tractography of the Probst bundle, and here you have the corresponding section. And all the images I'm showing of this case are in the new paper that just came out this month, I think. And the next thing was, okay, now we see the Probst bundles, but if they are really a heterotopia, then they should have the same intrinsic organization as the corpus callosum. And of course, you know the intrinsic organization of the corpus callosum because there is also here the fornix and the cingulum, and we can find these structures also in the Probst bundles. Well, you will say, well, this is not enough. Are you not able to quantify your results? Well, yes, we are, because there is a possibility to quantify the data of the diffusion tensor. We can measure a so-called fractional anisotropy, and we compared this fractional anisotropy of our Probst bundles with age-matched corpus callosa of fetuses, and there is really a great overlap. So the structural integrity and maturity of the Probst bundle is very similar to the corpus callosum. Well, I show you another case. What is this? Is this a dysgenetic corpus callosum? Dysgenetic because it is misshaped and here it is too thin, or is it something else? Well, if it is a corpus callosum, we should not be able to find Probst bundles because the Probst bundles are the corpus callosum. In this case, there were Probst bundles. So the structure we are dealing with cannot be the corpus callosum. It is something else. And it is a structure I have already mentioned before because it is a persisting hippocampal commissure. Now, we know already from experience from neuropediatric departments that a persisting hippocampal commissure in callosal agenesis might be associated with a better prognosis than a partial or callosal hypogenesis of another type. And why are we interested in knowing this? Because lately disorders of axon guidance have been defined, and for most of them a genetical background has already been found. For callosal agenesis, this background has not yet been identified, but geneticists tell us it is a matter of years when we will know the genetic background of these disorders of axon guidance. And when we will know that, we will have another little mosaic stone in being able to give a more accurate prognosis to these children. It is, of course, not the only influencing factor. The others have already been mentioned, like such as malformations of cortical development or complex malformations, but it might be a little bit of a mosaic stone. Well, I'm still with the same case because tractography was not the only thing we performed. Gustavo, you want to add something?
Yes. Actually, it should be the fractional anisotropy should be half, because there are not only fibers that go through. You have half of the axons that are there. So the fractional anisotropy need to be half the true anisotropy.
No, this is
because
the fractional. the quantification is not done as you think. The quantification is fiber density and myelination, and it is not the whole thing. Yes, it is another factor that we're looking at here. Sorry. But staying at the same fetus, we performed another test here. You see it was at gestation week 36, and here you see yellow dots. What do the yellow dots mean? The yellow dots mean that there is brain activity because we performed a so-called resting state MRI. And you can imagine that when there is brain activity, there must be some brain working, and where is no brain activity, well, there may be brain, but maybe it is not working. But I owe you an explanation, I think. So how did we do that? You know perhaps about functional MRI. It is called blood oxygen label dependent imaging, and it is based on the fact that when the brain is active, the oxygen consumption rises, and you can measure this difference in comparison with non-active brain regions. Usually, we do it in adults or in children when surgery has to be done and when we don't want that something is resected that has a function. So if something has to be resected that has a function in the motor area, we have finger tapping, and then we get these signals in the area, in the central area of the cortex. And now you will laugh and say, now she's telling us stories, because how will she tell the fetus to do finger tapping? Well, we don't, but there is another method, and this method is called resting state MRI. What is resting state MRI? It is the basic cerebral activity. Usually we tell the people, relax, stay awake, lie still, eyes closed, think of nothing. Well, it's what you are doing now. Your brain is still working, I can tell you, and we thought it was really a good working place description for a fetus. What the facts are that we get low frequency fluctuations, and under resting state conditions, these fluctuations are organized in networks. And what we wanted to show at first in Normas were those networks. Well, the first reviewers, in the meantime, we fortunately have already 3 papers published, say, how do you know the fetus is not sleeping? We know it. Because you know REM sleep, you see, and this is the sequence that supplies us also with an opportunity to assess eye movement. So we know when they have REM movements, and so we omit to take the data during the REM sleep. Okay, that is what it looks in adults. This is acquired during 1 hour of imaging and with cooperation. And this is acquired within 1 minute of imaging and without cooperation. But still, you can see the frontal network through the ages and an occipital network, for instance. And then we thought, well, we can only have activity where vessels are because the blood is flowing in vessels. So some vessels have to be there. And then we went to some of our postmortem studies we did at 7 Tesla, and of course we also know it from histology. But you see exactly in this region we have the penetrating vessels, so it really could be true that we get a signal from there. But for the reviewers, of course, this argument was still not enough, but fortunately, because we thought the activity came from the subplate zone. And you know, as I showed before, the subplate zone is the really functioning cortex of the fetus until gestation week 28 or 30. And fortunately, there was a paper where they exactly were able to elucidate such electric activity from the subplate zone, but not from the cortex. And they found spontaneous electrical activity, which may also account for the signal we see on our resting state images. And then we did another test. We put a so-called seed in the cortical plate, this is the lower row, and in the subplate. And when we put the seed in the subplate, there were more regions firing than in the cortical plate. So what we are looking at mainly is subplate activity. And in the meantime, we are also able to track the motor cortex down, the network that is responsible for motor functions, and to go back to callosal agenesis. Of course, we are messing around with malformations. And now look at this. This is complete commissural and agenesis, even with interhemispheric cyst. It's not a nice-looking brain, but still you have a lot of frontal and occipital activity. Gestation week 32. Here we have a fetus in gestation week 31 with a much nicer brain. There is much more brain, but we could not find as much activity as in the fetus before. At the moment, this is still experimental work, but we have the hope that when we get more experience, that we might be able to tell you something about brain function in these cases. This was the confusion of the commissures. Now, the confusions of the metabolic disease follows. Gestation week 34. The referral for MRI was because of slight ventriculomegaly. In red, you have the sick fetus. In green, you have a normal one, age-matched. Well, you see the ventricles are wider. What you also see is there are some subependymal cysts and germinolytic cysts, if you want. And if you look at the frontal cortex, there are way too many gyri and sulci compared to the normal one. So this is bilateral frontal polymicrogyria. When a neuroradiologist sees this, next look is at the kidney, of course. And then what we saw, it is blown up a little bit hard, we saw a lot of kidney cysts. Then we went to the liver, also very normal for a neuroradiologist, don't you think so? And we have a possibility to image the liver to get some insights into the content of the liver, whether there is a metabolic change in the sense of fatty tissue. And usually in these images, the liver should not lose signal, but in our fetus, the liver lost a lot of signal, so we knew the liver was not normal too. And then we looked back to the brain and we saw the white matter, and the white matter looked too bright. Well, this is only a subjective impression. Can we prove it? Well, of course we can, because we can do tractography, and if you compare it with the normal case on the outside, you will see that the forceps of the corpus callosum ends here and is not as beautiful as in the normal case, and so there is for sure an abnormality in the white matter, even also better displayed in this sequence, which is a type of a T1-weighted sequence. And then we applied proton spectroscopy. I will go shortly what we see. We see some peaks, and we have to interpret the peaks. We cannot choose what we want to see. We get some peaks. Choline is the measure for cell turnover, creatine here for energy supply, NAA for neuronal function, a marker, myoinositol is a marker of osmolysis, and then there may be peaks in these regions that they may be lactate, lipids, or macromolecules. And this is spectroscopy of our fetus. Just look here, high myoinositol, which is seen in gliosis, so it would be in keeping with white matter pathology, and there were peaks in the lipid lactate macromolecule region. So now we are able to make a diagnosis because we have bilateral frontal polymicrogyria, abnormal white matter, germinolytic cysts, lipid lactate macromolecules, kidney cysts, and a pathological liver. And what is here comes out is Zellweger syndrome. This is the pathological confirmation of everything. And every time I show this, they say, well, in the 36th week, why couldn't you do the diagnosis earlier? Well, the problem is we are dependent and we are biased by ultrasound. If there is no ultrasound abnormality suspected early, we won't be able to do an MRI. And of course, we could have done the diagnosis earlier. But if you have no suspicion, we also can't do anything. Okay, next case. Such cases were, of course, already shown by Gustavo in ultrasound, short bones, big head, small thorax, and you see in gestation week 23 gyri and sulci that don't belong here. The best method to look for skeletal dysplasia is not MRI, is not ultrasound, it is CT. This is postmortem CT showing you all the features of this thanatophoric dysplasia, but you can have the same pictures also prenatally and with a very low radiation dose. We are working on low dose protocols. And this is the postmortem proof showing you this, a pathological gyration in the posterior medial temporal lobe, and here the profile is also very pathological. You saw that prenatally, and this is the neuropathological. It is very bright. I don't know whether you see anything, but I think you get an impression of pathological gyration here. So all that was already shown by Gustavo. And now, of course, it is already in the literature. And there is one very good paper by neuropathologists that are describing the mutations in the fibroblast growth factor receptor 3 gene. And the findings in these cases, the brain findings, are temporal lobe enlargement, deep transverse sulci across the infromedial temporal surface. So it is not somewhere. It is in a distinct region in these cases, and hippocampal dysplasia. And why? Because the mutations of these genes would be responsible for aerial patterning. Therefore, you might see hippocampal dysplasia because they act exactly in this region. There is a lot of progenitor proliferation, and apoptosis, it's not enough. What does it mean? There are too much cells. If there are too much cells in the cortex, when the cortex grows, what happens normally? It falls. So it starts to fall even if it's premature. And from their data, they did this very nice graph showing normal development and the development in FGFR3 cases. And this is what the drawing they did. And it was not the results of their study, it was just a postulation. And what they wrote was this. This is just an assumption, because what they found out is that the genes were active already at gestational week 8. And so they were already active, and they thought we should see the consequences of this activity much earlier than gestational week 18. And what is the proof that this is true? The cases Gustavo showed, because they were much younger than gestation week 18, and there is also one case in MRI in gestation week 17 where you can see the same. Okay, this is nothing new, and the next case is also nothing new, because Rabbi told me he wanted me to show another case of, well, of what? You have to do the diagnosis because I've forgotten what it is, yes? Because this is already part of the quiz. Yes, Erich Hafner, who is also a level 3 sonographer, did this ultrasound. We did the MRI. There is no corpus callosum. You cannot see the eyes, but what we could see is big plexus cysts. It was a female fetus. And you see, here is a very strange cortex. Here is a very strange cortex. And here you have the plexus cyst. And here we tried to to correlate the ultrasound with the MR images. And all these cases looked the same. And when Eric saw what we saw, he went back to the ultrasound and he, of course, saw the same. So you don't need MRI to do the diagnosis because all these cases look the same. And this is, of course, Aicardi syndrome. And the last case I put in while watching Gianluigi's talk because I just wanted to tell you the radiological approach to holoprosencephaly. Well, this is holoprosencephaly. Do we need MRI to make the diagnosis? No, of course not. At that time, we called it semi-lobar. If you want to call it a lobar, also fine. I don't know. But of course, we try to use our modern methods to look what goes on inside this brain, because there is some brain left. So what is going on inside there? And of course, we perform resting state MRI. Big surprise. There is a synchronous bilateral frontal activity. But why? There is no corpus callosum. How can there be an integration of activity between the hemispheres? There must be an explanation for that, and because we really couldn't explain it, but then we got the explanation by doing trachography, because there is a hypertrophic anterior commissure. I'm showing you this as my last case just to show you what we are messing around with. It is far from clinical relevance right now, but perhaps in some years it will be. If you want to learn more, you know what I'm going to say, please visit me in Vienna.